Hello, my name is Blake Bextine, and I'm an associate professor of biology at the University of Texas at Tyler. I'm a molecular biologist, and I work within entomology and plant pathology. And the main focus of my project with Zebra Chip and the Specialty Crop Research Initiative is to study the population genomics of the insect and track the movement of potato psyllids throughout all the growing regions, potato growing regions in North America and Central America. And so as technology has developed over really the past 20 years, our genomic capabilities have changed drastically. And so we've been following this trend of collecting massive amounts of data genomically and finding ways to computationally approach these large data sets to gain understanding of genetics and genomics of the insects involved. And so we've been leading in the potato psyllid genome project. This is following a project that we've had with the Asian citrus psyllid also. And as we've moved through the genome project, it's given us quite a few nuclear markers and mitochondrial markers that we can use to phylogenetically understand how potato psyllids in these different regions are related, not just to each other, but related within the regions. So using most of the genomic information that we've had, that we've captured, we've gained quite a bit of knowledge of which populations of psyllids are related to which other ones. And so in some of our original work, we looked at two different biotypes. There's a third haplotype that's been found by another group. But we have documented that this central biotype is all the way through the central U.S. and has a range that goes down as far as Nicaragua into Central America. And we see a connection between these insects that we've collected in these locations all the way from as far north as North Dakota and as far south as Nicaragua. Now, similarly, we've seen a connection between insects collected along the west coast, California mainly, and those that have been collected in New Zealand. So we see a population correlation between the two. We use quite a few different techniques so that we can understand this better, and we're not relying on a single piece of information. So we've used nuclear markers, mitochondrial markers, and those have been developed because of our genome project. We've also developed metagenomic assays where we actually look at the bacteria and the genomes of the bacteria that are contained within the organism. And this is a really dynamic way to look at things because bacteria in an organism can be either obligate, which means that they have to be there, or facultative, meaning that they're transient and they come and go. So using those as population markers gives you a good idea of interrelatedness between different populations of insects based on the microbiota that's contained within them. So from these techniques that we've used, we've been able to describe connections within the central U.S. and also the west coast of the U.S. We've also seen immigration of west coast psyllids into the central U.S. and immigration of central biotype into the western U.S. So all that said, I think the world is quite a bit smaller than we really thought it was. And psyllid populations that are occurring in a different part of the world can be very important to the situation back home. So as a whole, we need to make sure that we look at these large populations and these large data sets and analyze them in a way that we can all help each other in understanding